Our featured speaker today is integral, central part of our community. Dr. Eric Schmitt is an assistant professor at Baylor College of Medicine and a genetics researcher, and he's given us some great talks before, and we're looking forward to another one. Let's welcome Eric to the podium.
Thank you. Okay. Let me know. So we all owe Aaron a big thanks because it was my intent to have you all sit here until I nailed this talk. Okay, today I'm going to talk about epigenetics, subtitle, Why It Matters What Your Grandmother Ate. And that actually leaves out a bunch. Not only does it matter what she ate, it matters when she ate it. And in the end, we're going to talk about Grandpa a little bit too. Okay, so what's epigenetics? Literally means epi is above, standing above genetics. And it's something that really hasn't gained much attention until recently. Epigenetics is one of many forms of non-Mendelian genetics. Mendelian genetics, I'll show you an example in a little bit. That's what you learned in high school, where you do your little Punnett squares and your crosses and figure out your dominants and your recessives and what the phenotypes are going to be. Epigenetics is a way of tagging genes. Usually what you're doing by tagging them is turning them off. And the tags don't change the underlying DNA sequence, but they do change the way the DNA is utilized. And that's really important because when you think about evolution, what evolution does on a Mendelian level is you have a population that has some variation in it, the environment changes, and those individuals who have the appropriate combination of genetic changes are favored and leave more progeny. That's a slow process. It turns out nature's come up with a fast process too for handling things, and that's what we're going to talk about today with epigenetics. So here's an example of Mendelian genetics, one that might surprise you because everyone's used to thinking of mutations in genes as being recessive things, but I picked achondroplasia here, where the mutation is actually dominant. So you've seen individuals who have dwarfism. The common form of dwarfism is achondroplasia. And an individual who has achondroplasia has a big A gene, the achondroplastic gene, and a little a, which is the normal stature gene. And the achondroplasia gene wins out, and the individuals end up short statured. So if you have a big A, little a genotype, you have a dwarfism phenotype. If you have big A, mom and dad both give the big A, that's actually a lethal condition. There's so little growth that the chest cavity, the chest volume is too small to support respiration. And if they each give the little a, two dwarfs can actually have a normal stature child. Happens quite often. In fact, happens 1/4 of the time, right? Or out of a live birth, the surviving births, 1/3 of the time. So that's Mendelian genetics. What's epigenetics? The older members of the audience will remember those M&M commercials. These cards are marked. They're a mess. They're a chocolate mess, and they've got fingerprints all over. The value of the card hasn't changed, but the cards are marked. You can tell what they are. And epigenetics is very similar to that. You've got a gene. The nucleotide sequence of the gene is the same. But what you've done is you put a tag on that gene. Typically, the best studied case is you put a methyl group, this little CH3 group, plunk it onto the cytosines. And those occur in what are called CPG islands, so cytosine, phosphate, guanosine. And so here's an example of a methyl group sitting on a cytosine being attached. And you put that on one strand, and then it gets marked, the other strand gets similarly marked, and that marks a gene usually to be shut off. There are other types of epigenetic modifications to the DNA. They're not always methylation. Sometimes there are these things called histones that are these protein complexes that you wrap the DNA around. If you wrap the DNA around real tight, you can't express the genes. If you loosen that complex up, you can read the genes and make the gene products. And so there are methylation changes that can occur to the histones, or acetylation changes that can occur to the histones that will allow genes to be expressed. We do this all the time, actually, in a reverse way. Males and females, males have an X and a Y chromosome, females have two X chromosomes. So for most genes, you need one copy of it, because males are getting by on one copy, and two copies is a dosage problem in many places. And so the way we deal with that is, for females during embryonic development, they randomly shut down one of the X chromosomes and only express one of them.
Oh. Yeah.
Okay, so what's the purpose of epigenetics? Well, we had to have it. Our genomes are littered with junk. Seriously, I mean, some of it is the fodder, the fuel for driving evolutionary change, but a lot of it is invasions that have happened into our genetic material. Sometimes it gets put to good use, but a lot of times it can cause problems. 45% of our genome is retroviruses and retrotransposons, basically infectious DNA elements that can move around in your genome and cause problems. And we need to shut those things down. And so one way we do it is by sticking these methyl groups on them to shut them down so they don't get expressed. Very important in evolution. Also, for us to be here, right? We're complex organisms. We're not single-cell organisms. We've got to have specialized cells. This is how you go from having a pluripotent stem cell gamete to a zygote, and then have that differentiate into all the different tissues you have. You've got to be able to shut genes off and just express the ones you want in any given lineage. And so this is one of the ways that you do that. There are also parent origin expression patterns out there that epigenetics are used for. There are some genes where you need to have a copy expressed from your mother and a copy expressed from your father to avoid disease. And everybody says, well, you always get one copy from your mom, one copy from your dad. Not true. Sometimes you can get both copies from your mother, none from your dad, or vice versa. And there are ways that this kind of thing can happen. A famous example of this is there's a spot on one of the chromosomes that if you have a deletion of the paternal copy of the gene, you get a condition called Prader-Willi syndrome, which involves behavioral issues and obesity, a condition called pica, where they're just so hungry for food, they'll eat non-food items. I remember seeing a patient one time, and the way we picked him up was police picked him up in his neighbor's house because he had broken into their refrigerator to eat. And the reason he had broken into the neighbor's refrigerator to eat was his parents had locks on their refrigerator to keep him from eating, just his insatiable appetite. Take that same region and instead don't delete the paternal copy, you delete the maternal copy, and you get a condition called Angelman syndrome, which are these sort of thin kids with severe mental retardation and behavioral issues as well, but very different looking presentation. You need one from mom, one from dad. That's why, for example, in humans, you don't see parthenogenic birth. If you have parthenogenesis, that's where, and you may have heard of turkeys and things like that doing it, an egg goes through a doubling of the chromosomes and it goes on and divides and produces an offspring, and that offspring is viable. In people, you have problems with that. And that's going to be as close as I get to a religious commentary in this talk. So one of the things epigenetics can do is get us a rapid response to the environment. And I want to show you that. And this data is absolutely startling. So I don't know if you're aware of this. Everybody thinks identical twins are exactly identical. And they're not. As time goes on, they have more and more differences between them. And there are various reasons why identical twins aren't identical. But one of them is epigenetics. epigenetic marks are put on stochastically. They happen random. Once you put them in there, you maintain them. There's enzymes that go in. You put this methyl group on one of the cytosines, the DNA molecule gets both strands marked. It's a CPG, so next to the G on the other strand's a C. You get all that marked, and there's enzymes that recognize that and maintain that mark. And when the strands divide, now you get this hemimethylated DNA. One strand's methylated, one strand isn't. The enzyme comes back in and makes them both methylated. So you maintain it down the cell lineage. Okay? So depending on what gene gets turned off, you get different clinical presentations. And so when you look at identical twins, they do differ in their epigenetic marks. Those differences increase over time. And if they're reared in different environments, or if you select twins who are discordant for some medical condition, the fraction of epigenetic differences is higher, which is what you would expect. And there's a nice review of those that I put up there for those who want to see more on that topic. This is a super cool slide. There's a guy here, and he's actually at Baylor College of Medicine, Robert Waterloo, who did these beautiful experiments on epigenetics. And what you can do is you can take mice strains and you can make them genetically identical. You just take the mice and keep back crossing them. So it's like a martini, is one example I read one time. You got your martini, you put your olive in it, take the glass and your cross, you get rid of half of the genes. So it's equivalent to taking the olive out of the glass, pour out half the martini, add more alcohol, mix it up, put the martini back. That's one cross. And you do that, do that, and do that. In very short order of time, the drink is pure alcohol with your marker that you want to track. And so you can do that with mice strains too. And so what they have is these strains of mice that are completely genetically identical. And there is one of these genes, one of these retroviral elements that affects expression in this mouse. And if that gene is on, the mice become obese. They have this nice brown, yellow coat color. They have obesity, diabetes, and cancer risk. If you take those mice and you feed their mothers a diet full of things that will promote methylation of the DNA, folic acid and some other things, then what happens is you increase the methylation and you go to a mouse that's a little smaller, a little patchier in color, until you get methylation the whole way cranked up and you get these brown mice that are thin body habitus and don't have the risk for obesity, diabetes, or cancer. All you've done is change the mom's diet. And it turns out that when you cross these mice, you can produce grandchildren, grandpups, that are thin and have avoided the risk for diabetes and cancer. But that is a little tricky. It turns out we reset our methylation pattern during gametogenesis. So as we make our eggs and sperm, we remove the imprints that are there and we make it pristine and put down fresh imprints. So to get that intergenerational, get the grand generation affected, you actually have to have that dye in place at the time the imprint's being established for the egg. So it hits not just their offspring, but the grand offspring. All right, does it happen in people? Yeah. There is a population in Gambia that had been studied quite some time ago. In Gambia, they have a rainy season and a dry season. And in the rainy season, it's hard to get your food. And so the rainy season is known as the hungry season. Those conceived in the hungry season have 10 times increase in premature death. It's thought to be caused by immune dysfunction. So Bob Waterloo again had worked on a group of these individuals looking for genes that might be epigenetically modified. And he was pretty tough about eliminating a lot of things. So he probably threw a lot of real genes that could be epigenetic. But he really wanted some good solid cases. And he got it down to about 100 candidates, and then out of that he followed, I think it was 6. And what he found was that there is increased methylation for some genes during the rainy season, and it behaved like one would expect, meaning it was stochastic. So you had this collection of genes. Not everybody had methylation of all of those genes. If you looked at twins, they could be discordant, but they looked at autopsy tissues too, and so they saw that it was true to the lineage. Once that methylation imprint was put on the gene, it got passed down that lineage, and they looked at endoderm, ectoderm, mesoderm, and showed that the imprint was maintained. The expectation they had gone into it with was that when they looked, they thought that it would be, okay, they're poorly nourished during the rainy season, they're going to have low methylation because they're not getting nutrients in it. Instead, they found just the opposite. That's when the methylation occurred. And at first they were puzzled by it, and then what they realized was that during the rainy season, during the hungry season, the people were eating a lot of foliage. Foliage is folic acid. Folic acid is the stuff that drives the methylation. So it all fit beautifully, actually. And here's another example of this. This talk's going to go by real fast here, I think. This is a province in Sweden over Kalix. Data is absolutely stunning. The original study that was published was back in 2001. So in this portion of Sweden that was rather isolated, where you couldn't get train service or boat service in to bring in fresh food, they had some really bad harvests. And there's historical data that goes back 150 years. And so what they did was they looked at that data and identified years of starvation versus years of feast, and looked at what the outcome was for the children who were born during that time. What they actually looked at was not when they were embryos, but when they were in their slow growth phase. So their slow growth phase was just before puberty, and they figured that that nutritional status was involved in producing the imprints on the sperm in men. So they were looking at men initially. They did branch out and look at women at one point. So what they looked at was when Grandpap had his slow growth period during years of famine versus when he had it when there was plenty of food around. So they had different time periods, and they compared those grandfathers and looked at what happened, and they looked at the male lineage. And what they found was the grandchildren of Grandpaps who were nutritionally starved during their slow growth period had a life expectancy that was 32 years longer than the individuals who were fed well. 32 years. This wasn't some small number of cases even. They actually sampled 50% of the cohort from the records that were available there. And they had it broken down into groups that had feast, famine, or something in between. And the data held up. But, and these are large numbers, are statistically significant. And that sort of got everybody's attention, what's going on here. So they came along later to look to see a little more detail what was going on. And when you looked at grandfathers who had famine during the slow growth period, their children actually had lower risk for cardiovascular disease. And you could actually see the same thing when you looked at grandmothers who went for for the opposite. The grandmothers had feast during their slow growth period. The kids had lower cardiovascular disease. So it's just the opposite between males and females. As though the coffee you're getting from mom and the coffee you're getting from dad are marked differently. Just like I was telling you that Prater Willie Angelman story. There is a candidate locus genetic gene that does sort of fit the bill. It needs to be studied intensively to figure all this out, but there's insulin-like growth factor 2 gene that gets paternally imprinted that would be a good candidate for this because it sets you up for diabetes and heart risk. And when you looked at the grandchildren, actually it's almost like there's an oscillation here, so they were lower risk for cardiovascular disease, but when you looked grandfather to son, their grandkids showed a 4-fold increase in diabetes relative to individuals who have gone through the family phase. This one is relatively new. It needs to be reproduced. I remain skeptical until I see this demonstrated in a repeat study. But I looked at the paper and the data looked pretty good. We're talking about how epigenetic marks can get you to respond to the environment in a quick way. One of the thoughts was, if you're born in lean years, you're going to hang on to your food, you got to be careful about what you do with it. You're affecting the utilization of whole pathways to conserve energy. This group looked at memories. So there's this compound, acetophenone, which I'm trying to remember what odor they said it smelled like. It was cherry blossom odor, so a cross between cherry blossom and I forget what the other compound was, but it had a characteristic odor. And there's actually a receptor for this chemical in the mouse nose. And they did a Pavlov experiment where they conditioned the mice by exposing them to the odor and then applying shocks to their feet. And the mice learned that they didn't like the odor. Big surprise. But what was really shocking was that when they looked at the offspring responded to the odor. They responded by shuddering when they were presented the odor, even though they had never had the paired conditioning. They looked at the gene. There's a specific odor receptor gene, this olfactory 151 gene, and they looked at that gene in the pups, and that gene was undermethylated, meaning it was being used big time. And when they looked, the expression of that gene was they had a 200-fold increase in sensitivity to the odorant versus the prior generation. So they turned this gene way on, presumably in the conditioned mouse, and they managed to affect the germline, get it turned way on in the germline. That imprint got passed to the pups, is the interpretation here. And so the pups are expressing that odor receptor at high level. So you present this odor and they're like, whoa, I know that odor. What's surprising is that there's a shudder response. I don't know how you get from being able to pick up the odor to being able to respond to avoid the odor, and how that's getting transmitted, and the authors don't know either. So we really need to see that all worked out. But they were able to show this effect also through IVF. So they took sperm from the mice that had been Pavlovian conditioned and used those to do in vitro fertilization of eggs. And those pups also showed this change in their gene. Okay, and again, it's not the gene itself, the DNA code of the gene isn't different. It's just marked to be used differently. They also showed that the experiment works if they condition mothers, so it's not unique to the fathers. But again, I caution that all of this needs to be reproduced, and they need to exclude other explanations. For all you know, there's some researcher in there who's not doing a double-blind experiment and going, okay, here's the other. Okay, now we're really going to watch this mouse. Yeah, twitch. You can mislead yourself. And, these are smart researchers. I think they didn't do that, but you want to see other groups challenge it. So this is an example of where epigenetics has gone. And it's basically from the recognition that we saw it, it's ended up everywhere. Fetal versus placental growth control is one issue. So genes you get from your father tend to be promoting growth of the placenta. Genes you get from your mother tend to be suppressing growth of the fetus, protecting the mother against swapping a baby that she can't deliver. I mentioned the X chromosome inactivation earlier. So females shut down at random one of their X chromosomes. If you've ever seen the tortoiseshell cats, that's a good example of that. So the color gene is on the X chromosome, so they either express the yellow color or the black color. And the genes are shutting down at random, so you see which gene is on based on the patches. And so all the women here in the audience are actually a mosaic of two different sets of X chromosome expression. And sometimes, if there's a genetic disease running around in the family on the X chromosome, women can manifest if they're unfortunate enough to shut down the functional copy of a gene and leave the non-functional copy of the gene intact, being expressed. So if the one you didn't shut down doesn't work, then you're stuck with how to work the gene. Stem cell differentiation, so I mentioned that as we develop as an embryo, we have to shut off particular genes in different pathways so that genes don't produce disease. And there's actually these respond, sometimes the genes are just like completely shut off and there's no way to use them. Sometimes they're just getting cues from the environment to tell them what genes to use. And you shut things off based on the environmental cues that you're getting. So there's a tumor actually that occurs in the ear that's a rather interesting one. So if you get some cells from the outer ear into the inner ear canal, those cells can lose their growth suppression and start to grow, and it'll actually produce a benign tumor in the ear canal that can affect hearing. Epigenetics are important because if it's not in place, you liberate all these transposable elements, these viruses, and they move around. If they land inside a gene, they can kill that gene. Sometimes, and this is part of what drives evolution, sometimes they land in front of a gene, they start using their promoter to express their gene, but they also start expressing whatever's next to them. And they can express silent material, or they can boost the level at which some genes are on. And that's played a role in our evolution. It's thought that epigenetics is involved in our environmental response. Stress is one. So there is data out there that for abused infants, they actually have particular genes that get methylated, and it affects their health for life. They shut down particular genes as a response to the stress, and they're just off for life. Innate behaviors, I mentioned this learned conditioned response. I mean, imagine if you had to rely on evolution to select your avoidance to the cherry blossom, and how much of the population would have to crash till you actually had an individual who had a successful combination of genes that allowed it to leave progeny and not get killed off if that had been a fatal event. But here, in a generation, actually in the exposed generation, in the exposed individuals, there was a change in response. So it's the way of using the genome in a fast response. And it plays a role in things like obesity, diabetes, in vitro fertilization. One of the steps often is culturing cells. They take the embryo that they form, they grow it up to a certain cell stage, especially if there's like a genetic disease and they want to select for unaffected embryos. They'll pull a cell off and they'll do some genetic testing on the cell that they biopsy and determine whether the embryo is affected or unaffected. Those are sitting in culture for a while. But one of the observations that came out of this was that there's actually an increased frequency of Angelman syndrome and Prader-Willi syndrome in babies who are conceived through IVF. And the reason they think it was happening is the culture conditions were not appropriate. They were getting changes in methylation of those genes. So now it's one of the things that they have to watch for in the culture. And then cancer progression. How you use your genes is affected by the methylation pattern. It turns out cancer being a selective process, the cells that get the appropriate changes that allow them to go through unrestricted growth are the cells that take over in the body. And so a lot of what goes on in cancer is shutting down genes that would normally halt growth. And back to epigenetics. And then good old-fashioned aging. We've got you should look at aging as a disease. It's not normal. We're used to thinking of aging as being just normal. That's what we all go through. But you should think of it as a disease. We have these genes that allow us to produce these tissues that are wonderful at one point in our life. And somewhere along the way, things pumpkin. You turn into the pumpkin. It just your stagecoach is gone. You just end up degrading the use of your genes. And there's a variety of reasons for it, but epigenetic marks are part of it. In fact, I don't know if any of you ever heard about telomerase, but the telomeres are special ends at the ends of your chromosomes. They're just hanging in the breeze. And so you put these special caps on them to protect the DNA. You can't replicate. When you replicate a strand, you gotta go out on this strand, you gotta come back on the other one. Well, coming back on the other one, you're off in space somewhere. You've got nothing to come back from. So nature has created this special end to the molecules. But as we get older, every cell division, these telomeres get shorter and shorter and shorter. You lose because you can't prime out there. And eventually they're gone. And eventually you start losing gene function. And it may be a rigged system. We may be doing it for a purpose. It's one way you can control cancer. Because if you only get away with so many cell divisions before you start chewing up your chromosomes, then that cell commits suicide. Well, okay, you get a cell that becomes cancerous, it loses its telomeres, gets gene damage, commits suicide, cancer cured. And we probably all get cancers every day, and get rid of them safely. When you look at cancer cell lines, and one of the things they do is they turn this gene on, this telomerase, to keep fixing the ends of the chromosomes. And again, there are epigenetic changes that occur to drive all of that. So epigenetics is shown up everywhere. It just does some astounding things. I mean, who would have ever thought that depending on what grandpap ate, you could have a 3-decade difference in your lifespan. But that's what the data is telling us. So one needs to really be mindful about what our condition is during our life, actually, because these cells expand cloning, but also during our reproductive years. And you got to think about what are the consequences of delayed reproduction. As you wait and wait to have your babies, are you accumulating bad epigenetic changes that are going to get passed to the baby? So, with that, we are done. So, thank you.